All right, hello guys. Today we're going to be taking some notes on data and data analysis. In your notebook, in the table of contents, the title is going to be Data Analysis and the page numbers are going to be 13 through 14. This time we're just going to do notes. We are not going to do an output page. You're going to want to set up pages 13 and 14 for Cornell notes, so set up your two columns. At the top of the page, you should have the title, again, Data Analysis. Below that, you should write the LEQ. How can we draw valid conclusions from the data that is collected in an experiment? If you are an AVID and you need to write a citation, it can either be teacher or PowerPoint. And make sure on page 14 you leave some space for your summary. If at any point during this video you need to hit pause so that you can catch up on the notes, feel free. Just make sure you are keeping an eye on the time so that you don't run out of time. All right, so data. Data is very important because all of science is based on evidence. Evidence is data. Now, data is any type of observation or measurement collected during an experiment. Measurements can be just about anything. It depends on what the experiment is and what's being done, what the question is, what the independent variable is, what the dependent variable is. Usually in here it's going to be some sort of measurement, some sort of quantitative observation using numbers. One thing that's important is that data is as accurate and precise as possible. We want to be as careful as we can when we're collecting data because we want to make sure that we are being correct. We want to try our best to avoid any errors. Now, accuracy and precision are two similar things. Accuracy is how close a measurement is to the true or accepted value. And precision is how close a group of measurements are to each other. So for example, with accuracy, let's say we're trying to find the mass of an object. And I know that the object is, say, 5 grams. Now, if you measure it, and you measure it as being 4.8 grams, you're very close to accurate. You're close to the true value of it. But if you measured it and you got 10 grams, that's much less accurate than the first measurement. Now, when you're collecting a set of data, again, with that example of the object being 5 grams as its true value, if you collect a set of data and it's all within, it's all very close to each other. Maybe your range is 4.7 grams to 5.2 grams. That's a very small window of measurements, so they're all very close to each other, so that would actually be a very precise set of data. All right, we've got quite a bit of vocab on this slide. So how do we analyze data? Anytime we collect data, we need to look at it in order to figure out our conclusions. We need to answer the question that our experiment is on. So a few ways we analyze data. We use a few things called the measures of central tendency, and that's the mean, median, and mode. Mean is the average, which we did during our bubble lab. You did 5 trials, and then you calculated an average of those trials. The median is the middle number of an ordered set of data. And the mode is the number that appears the most in a set of data. Now, range is the difference between the greatest value and the least value in a set of data. So if we have a set of data that our lowest value is 5 and the greatest value is 10, so we've got a range of 5 in between there. So that'd be 10 minus 5. That's how we calculated the range. Percent error is a little bit more complicated. We won't really be working with it, but it is something that is used a lot. And basically what that is a calculation of a percent difference between the known value of a substance and what it was measured as in an experiment. So again, that's not something we'll really work with much in here because it is much more complicated to calculate. But it is something to definitely at least know what it is. Now, error. Errors happen. It's very common. If an error happens, it doesn't necessarily mean that it's a bad experiment or anything like that. It does happen. Sometimes they're caused by people making mistakes. Sometimes they're caused by equipment not working properly. But errors can cause something that is called anomalous data. And what that is any sort of data that does not fit with the rest of the data set. So for example, in the data chart off to the side of this slide, the adult male Florida black bear masses, if you look at that, there's 5 different bears and their mass is listed. Now, most of them fall somewhere between the lower 300 kilograms and upper 200. But there's one that is way bigger than the others. Bear D has a mass of 825. That's well over twice the mass of the second largest bear, or the second highest mass. So that is an example of anomalous data. It's something that it doesn't fit in with the rest of the data set. So anytime someone sees anomalous data, they need to look into what caused it. They might look for something like an error. So for example, maybe in this case, the person wrote down the mass wrong. Maybe it was supposed to be 285 kilograms instead of 825. Maybe they just mixed up the 8 and the 2. Maybe something was happening with the equipment and it was malfunctioning. Maybe, who knows? There's a lot of different things that could have gone wrong there. Now, sometimes the anomalous data isn't caused by an error. Sometimes it could be caused by some other variable that we didn't know about. So looking into the cause behind that could possibly lead a scientist to making a new discovery. So anomalous data isn't always a bad thing. It just happens. And that's it for your notes. So you need to make sure that you are writing a summary. That is part of the Cornell notes. Very important. Your summary should be written below your notes on page 14. And it needs to be at least 3 sentences long. Sum up what we talked about in these notes, and try to answer the essential question as well. And that's it.